Hello everybody, I'm Dr. Daqin. As a chairman of our IVF team, I will make a brief introduction of PGT-A because it is frequently asked about in our daily practice of IVF clinic. PGT is the abbreviation of pre-implantation genetic testing. A is the abbreviation of Aneuploidies. In civilized countries such as United States, Japan, and Taiwan, the childbirth of advanced women aged 35 to 39 continues to increase, as even more advanced women over 40 years old also increase their possibility of childbirth due to infertility treatment. Due to the advanced age, the egg quality of the elder woman becomes worse. The rate of chromosome anomalies becomes higher. Therefore, they are prone to have chromosome abnormal babies and subsequent abortion. Therefore, in IVF, we have the technology of pre-implantation genetic testing, PGT, to check embryos' chromosome abnormalities, including abnormal number of chromosomes and the fragment deletions, etc. And PGT-A is designed to exclude the aneuploidy of the embryo. And the proper selection of produced embryos is expected to increase the chance of IVF success. There are three kinds of pre-implantation genetic testings, PGT, used in IVF selection of embryos. PGT-A for aneuploidies, we call it PGT-A. PGT for monogenetic or single gene defects, we call it PGT-M. And PGT for chromosome structure rearrangements, we call it PGT-SR. PGT-A is most frequently used technology in these three PGT. How we do PGT-A? When the embryos is produced after IVF, we collected several cells, or we call them blastomeres. In PGT-A, we check whether the tested embryo have correct numbers of chromosomes. The screened embryo have a correct number of chromosomes and are the most likely to implant and result a successful pregnancy. It also reduces the chance of having a child with extra or missing chromosomes, such as Down syndrome. How we do PGT-A? A skilled embryologist removes one or more cells from each embryo. The cells are sent to a laboratory to examine the number of chromosomes in the embryos. We test the embryos by counting the 46 chromosomes to detect extra or missing chromosomes. We call this kind of a normal embryo aneuploidy. We select the embryos without extra or missing chromosomes. That means embryos without aneuploidy. To embryos transfer, therefore we call it PGT-A. How we perform PGT-A? There are several technologies to check aneuploidy of these embryos. For example, real-time PCR, FISH, CGH, and most advanced NGS. In our hospital, we do NGS-based PGT. NGS is an abbreviation of next generation sequencing technology. refers as NGS, also known as high throughput sequencing technology, can sequence hundreds of thousands to millions of DNA molecules at a time to read and mark of long and short DNA molecules. It belongs to the second generation of gene sequencing technology. GNS can detect a variety of genetic change, including point mutation, gene copy number changes. In GNS-based PGT, we can not only detect aneuploidy of this embryo, but also the percentage of the mosaicisms in test embryos. In the right side of the screen, there are two results of PGT-A. In the above, this embryo has extra 1 chromosomes in both 18th and 21st chromosomes. Comparing to the lower picture, it obviously has normal chromosome in this PGT-A. Who need PGT-A? PGT-A technology in IVF is indicated in A. Advanced age. Women in her late 30s and beyond should consider this option, especially those in her 40s to have IVF. B. Habitual abortion. Those who could have pregnancy no matter in IVF or not frequently experience spontaneous abortion. C. Repeated unsuccessful IVF. Those who experience repeated IVF failures without apparent other abnormal findings in her IVF. The advantage of PGT-A in IVF: increased pregnancy rate, decreased abortion rate, increased healthy baby rate, selection of proper embryos to transfer, reduced multi-pregnancy rate, reduced number of IVF to success, reduced total cost to have a healthy baby in IVF. Because that, if you are 38 to 40 years old, even in the blastocyst of your IVF embryos, there will be about 65% to be aneuploidy in the blue line of the pictures. Therefore, you will have a high rate to have spontaneous abortion in the orange line in the picture. But in PGT-A IVF, we can reduce the risk of aneuploidy to 0.2 to 3% in the red line of the bottom picture. Therefore, PGT-A IVF can increase your pregnancy rate and ongoing pregnancy rate. In those having high risk of unsuccessful IVF, as in advanced age or repeated IVF failure or repeated abortion, we call it habitual abortion. PGT-A IVF can reduce your total cost to bring a healthy baby home. In conclusion, PGT-A IVF is indicated in 1. Advanced age, 38 to 40 years old and beyond, especially those are much beyond 40 years old. 2. Habitual abortion, when we highly suspect its abortion is due to chromosome abnormality. 3. Several IVF failure, when there are no special cause to induce her repeat IVF failure. Abnormal embryos should be excluded to achieve a successful IVF. 4. When you have several embryos, but it is very difficult to find good embryos to transfer or it is difficult to find a proper embryo to transfer, PGT-A may serve you to find a proper embryo. Finally, it is the end of PGT-A introduction. If you have any question, you can contact with us. Thank you for your participation. May you have a good day. Peace out.